T-cell activation is triggered by the presentation of microbial or tumor-associated antigen to naive T-cells by antigen-presenting cells, or APCs. There are two basic categories of antigen-presenting cells: professional and non-professional or atypical. Dendritic cells, macrophages, and B cells are all considered professional APCs. Atypical or non-professional antigen-presenting cells include mast cells, epithelial cells, basophils, endothelial cells, fibroblasts, eosinophils, and tumor cells. Professional APCs are highly efficient at taking up and processing antigen. They also express high levels of MHC class II molecules and co-stimulatory molecules, which are required for optimal T helper cell activation. Atypical or non-professional antigen-presenting cells typically lack constitutive MHC class II expression and are less able to induce the activation of naive T helper cells. Antigen-presenting cells take up antigens, process them, and present the processed antigens to T cells in the context of MHC molecules. Antigens coupled to MHC molecules are recognized by T cells via the T cell receptor, which, in conjunction with other co-stimulatory interactions, then promotes T cell activation. Antigens recognized in conjunction with MHC class II molecules result in the activation of CD4-positive T helper cells, whereas antigens recognized in the context of MHC class I molecules activate CD8-positive cytotoxic T cells, or CTLs. Activation of T cells by antigen-presenting cells is a complex process orchestrated by multiple signaling events. Recognition of the MHC antigen complex, along with subsequent signaling via the T cell receptor, is a required first step for T cell activation. However, antigen recognition and T cell receptor signaling alone are not sufficient for T cell activation. Indeed, T cells that only receive a signal from the T cell receptor become unresponsive or undergo apoptosis. The second signal required for T cell activation is provided by interactions between co-stimulatory molecules, expressed by the antigen-presenting cell and co-stimulatory receptors expressed by the T cell. An example of a co-stimulatory interaction is that between the co-stimulatory receptor CD28, expressed by the T cell, and the co-stimulatory molecules B71 and B72 on the surface of the antigen-presenting cell. Accordingly, a combination of two signals based on antigen recognition and co-stimulation are required for effective T cell activation. After activation, naive T cells become blast cells that proliferate by clonal expansion and then differentiate into either memory or effector T cells. Activated effector T cells are relatively short-lived and carry out cell-mediated antigen-specific responses to control pathogens and respond to transformed cancer cells. Memory T cell subsets have the potential for long-term survival and are mobilized in response to re-exposure to cognate antigen. A third signal, essential for the final effector function of activated T cells, involves the release of cytokines by the antigen-presenting cell. These cytokines bind to cytokine receptors on the T cell, resulting in differentiation of the T cells into a particular effector subtype. By creating the right cytokine environment, antigen-presenting cells are able to orchestrate an appropriate T cell response.